Hello everyone and welcome to our channel, Mootechie. A special welcome to all the dairy technologists and students of food science and related fields. Today we're diving deep into the upstream processes of fermentation, a crucial foundation in food and industrial microbiology. Our session begins with an overview of the upstream process. This involves all the steps before the actual fermentation begins. The key steps include screening industrially important microbes, preserving them, improving the strains, developing the production media and inoculum, and ensuring proper sterilization. Let's talk about the first step, screening industrially important microorganisms. The task of discovering new microbial compounds or improving known strains is quite challenging. Industrially important microbes include bacteria, actinomycetes, fungi, and algae. They are found everywhere, but the most common sources are soil and lake or river mud. The screening strategy has three important stages: isolation, preservation, and improvement of commercially important strains. When choosing an organism, we consider several criteria. The nutritional characteristics are vital. The organism must be able to use the ingredients in the medium to produce the desired product. The optimum temperature matters too. Using an organism that thrives above 40 degrees Celsius can significantly reduce cooling costs in large-scale fermentations. We also consider the organism's reaction with the equipment, its stability, and its ability to be genetically manipulated. Productivity is key, meaning the organism should efficiently convert substrate into product and give a high yield per unit time. Other factors include easy product recovery, high yielding and stable biochemical characteristics, not producing undesirable substances, and being easily cultivated on a large scale. The ideal isolation procedure starts with an environmental source, like soil, which is likely to be rich in the desired microbes. We can use selective pressure during isolation, for example, by using specific substrates or conditions to encourage the growth of certain types. If selective pressure isn't possible, we might target a known microbial group, like Streptomyces for antibiotics, which frequently produces useful compounds. The goal is to exclude common microbial weeds and encourage novel types. Advances in taxonomy and molecular biology help design more effective screening tests. Now we move to primary screening. This step allows us to detect and isolate microorganisms with potentially interesting industrial applications. It helps us find a few microbes with real commercial value, but it doesn't tell us much about their production yield potential. For example, to screen for organic acid producers, we can incorporate a pH indicating dye, like neutral red, into a poorly buffered agar medium. Microbes that produce acid will change the color of the dye. Another method is to add calcium carbonate to the medium. Acid-producing microbes will create a clear zone around their colony as they dissolve the carbonate. However, these methods don't identify which specific acid is produced. Colonies showing potential are immediately purified and stored as stock cultures for further testing. For screening antibiotic producers, a simple technique is the crowded plate method. A soil sample is diluted and spread on a plate to get hundreds of colonies. Colonies producing antibiotics will show a zone of inhibition in the agar around them. This colony is then subcultured and purified. The purified culture is tested to find its microbial inhibition spectrum, or MIS, which shows what types of microbes are sensitive to it. But we must be cautious. An inhibition zone might not always be due to an antibiotic. It could be from a marked change in pH or rapid nutrient depletion near the colony. So further testing is needed to confirm antibiotic production. Using a specific test organism can improve this screening method. Next we have secondary screening. This further sorts the microorganisms from primary screening, keeping those with real industrial value and discarding others. It can be done on agar plates or in flasks and small fermenters, and can be qualitative or quantitative. Secondary screening should evaluate the true potential of a microorganism for industrial use. It must determine if the microbe produces a new chemical compound. It should reveal critical requirements like specific pH, aeration, or other needs for both growth and product formation. This stage also detects genetic instability in the culture. It shows if certain medium components are missing or toxic to growth or product accumulation. It helps determine the product structure, whether it's simple, complex, or macromolecular. Secondary screening assesses the product's chemical stability and solubility in various solvents. It checks for physical properties like UV absorption or fluorescence, which are useful for detection and analysis. For some products like antibiotics, it's crucial to determine if there is any toxicity to animals, plants, or humans. It also reveals if the product exists in more than one chemical form in the broth and if it is optically or biologically active. Importantly, it shows whether the microbe can chemically alter or destroy its own product. All this information helps plan further research on the microbe and its fermentation process. Now let's discuss strategies for isolating these important microbes. We can exploit microbial diversity by searching neutral environments for strains that produce valuable products. The first stage is isolation, which involves getting pure or mixed cultures and then assessing which ones carry out the desired reaction. Sometimes, the isolation procedure can be designed to encourage the growth of producers right away. Other times, organisms are isolated first and producers are recognized later. Remember, the chosen isolate must carry out the process economically, so selection is a balance between the organism's productivity and the process's economic constraints. Preserving the isolated cultures is critical. The goal is to eliminate genetic change, protect against contamination, and retain viability. One method is storage at reduced temperatures. Cultures on agar slopes can be stored in a refrigerator at 4 degrees Celsius or a freezer at -20 degrees Celsius with subculturing every 6 months. Subculturing can be extended to 1 year if the cultures are covered with mineral oil. For long-term preservation of cells that don't survive freeze-drying, liquid nitrogen at temperatures between -156 and -196 degrees Celsius is ideal. This technique involves growing the culture to its stationary phase and then resuspending the cells in a cryoprotective agent like 10% glycerol. We can also store cultures in a dehydrated form. Dried soil cultures are common for sporulating organisms. Lyophilization or freeze-drying is another excellent technique. The culture is frozen and then dried under a vacuum, causing the water to sublimate. Cultures stored this way can last in a refrigerator for up to 10 years. Moving on to strain improvement. Wild-type microbes usually produce very low amounts of a compound. To increase productivity, we use improvement strategies. Traditional methods involve random mutagenesis and selection. Selected strains undergo repeated cycles of mutation and selection, often leading to a large yield increase after several cycles. Yields can also be improved by optimizing the culture medium and growth conditions. However, this is limited by the organism's genetic potential. To break that limit, we must modify the genome. The techniques used to genetically modify strains for higher production are called strain improvement or strain development. Genetic modification can be achieved by selecting natural variants, induced mutants, or recombinants. Recombinant DNA technology is particularly powerful, allowing the production of heterologous products and further increasing yields of conventional products. Now let's explore the media used for industrial fermentation. An ideal production medium has several key characteristics. Its chemical composition must be suitable, providing a source of carbon, nitrogen, growth factors, and mineral salts. For some fermentations, the medium should supply precursors to get better yields of the desired product. Buffering capacity is essential to maintain the optimum pH range. Acidic or basic compounds produced during fermentation can change the pH. Buffers like calcium carbonate can be added. Media with proteins, peptides, and amino acids have good buffering capacity near neutrality, as do phosphates. Avoiding foaming is crucial, as foam can lead to contamination and other issues. Defoamers, like hard oil mixed with octadecanol for penicillin, are used. They can be added before or after sterilization or during the fermentation. The ideal medium should be non-toxic to the culture or product formation. Consistency is important for aerobic fermentations. Liquid media allow air to diffuse under agitations. The medium should not be viscous, as this prevents air from penetrating the interior. Some media conditions help check contamination. For example, low pH in citric acid production using Aspergillus niger avoids contamination, and such media can be sterilized at lower temperatures. Product recovery is a major consideration. Medium components should make separation and extraction of the product easy and cheap. Finally, the raw materials needed must be readily available in large quantities at a reasonable price. A wide variety of raw materials are used in industrial fermentation, often preferring crude, economical sources, many from agricultural products. Let's look at saccharine materials, which are sugar-based. This category includes sugarcane, sugar beets, molasses, and fruit juices. Molasses is a byproduct of sugar refining. Cane molasses is about 95% fermentable sugar and is rich in vitamins like biotin. Beet molasses has limited biotin, so cane molasses or other biotin sources are added for yeast fermentations. In India, cane molasses is widely used in the alcohol industry. Fruit juices, like grape juice containing glucose and fructose, are used as carbon sources, for example in wine production. Cheese whey, a byproduct of cheese making, is a major waste product. It contains lactose, nitrogenous substances, vitamins, and inorganic salts. It's used in lactic acid production and single-cell protein or SCP production. Next are starchy materials from cereals like wheat and maize, or roots and tubers like potatoes. These require pre-treatment, either enzymatic or chemical, to convert the starch into fermentable sugars. Cellulosic materials are complex carbohydrates. Cellulose is a polymer of glucose. These materials like sulfite waste liquor, wood molasses and rice straw require pre-treatment. Sulfite waste liquor from paper pulp production contains hexose and pentose sugars. It's used to produce ethyl alcohol with Saccharomyces cerevisiae or to grow Torula utilis for animal feed, but toxic sulfur dioxide must be removed first. Wood molasses is produced by acid hydrolysis of wood, yielding fermentable sugars. Rice straw is a poor quality animal feed, but can be used in silage production, SCP, and mushroom cultivation. Hydrocarbons and vegetable oils are also substrates. Hydrocarbon mixtures are relatively cheap and are used to produce single-cell protein with yeasts like Candida. Vegetable oils classified as oleic, linoleic, or linolenic, based on unsaturation, can be used as antifoams or as a carbon nutrient source. Now for nitrogenous materials. Corn steep liquor, or CSL, is a concentrate from corn steeping. It was crucial for penicillin production and is valuable in many fungal antibiotic fermentations. Soya bean meal, left after deoiling, is a complex nitrogen source used in streptomycin production. Pharma media is a powder from cottonseed embryos. It contains 56% protein and is used in tetracycline production. Distillers' solubles come from alcohol production. The residue is concentrated and dried, supplying nitrogen and B vitamins. Other materials include groundnut meal, fish meal, and yeast extract. Precursors are substances that improve yield or quality by being incorporated into the product molecule. Examples are phenylacetic acid for penicillin G and cobalt for vitamin B12. Inducers like specific substrates can trigger enzyme production. Repressors are used to control culture metabolism. For example, media allowing restricted growth can shunt carbon to product formation instead of growth. Slow utilizing nitrogen sources like soybean meal can avoid nitrogen metabolite repression in streptomycin production. For gluconic acid production with Aspergillus niger, the mycelium is moved to a nitrogen-free medium to force product formation. Antifoams are surface active agents that reduce foam stability. An ideal antifoam acts fast, is not metabolized, is cheap, heat sterilizable, non-toxic, long-lasting and effective at low concentrations. Examples include stearyl alcohol, silicones, and various oils. If antifoams affect oxygen transfer, mechanical foam breakers are an alternative. Finally, we come to inoculum development. The main goal for bacterial processes is to decrease the lag phase in the production fermenter. The inoculum size and physiological state are critical. Bacteria should be transferred while still metabolically active. For sporulating bacteria, using an inoculum with many spores leads to a long lug phase. For protease production, a 5% inoculum of thermophilic bacillus in the logarithmic phase is used. A two-stage program for Bacillus subtilis involves growth on solid or liquid medium, then transfer to a seed vessel for more generations before production. Using a large active inoculum in a medium similar to the production vessel eliminates the lag phase. For vitamin B12 production from Pseudomonas denitrificans, a specific pilot scale program is followed. Acetic acid bacteria are sensitive to oxygen lack, so an active inoculum is essential. Often 60% of a batch is removed and replaced with fresh medium to start the next batch, but this risks strain degeneration and contamination. Strain stability is a major concern with recombinant bacteria. For E. coli biotin fermentation, using stationary phase cells as inoculum improved plasmid stability. In lactic acid fermentation, lactic acid inhibits the producer, so techniques like electrodialysis are used to reduce lactate in the inoculum for better quality. For anaerobic processes like acetone-butanol fermentation with Clostridia, a multi-stage inoculum program is used. A heat-shock spore suspension is grown through several stages in molasses medium. Using a very small inoculum requires perfect conditions to avoid contamination and long lag phases. Now let's look at inocula for yeast processes. Yeasts are used in brewing, baker's yeast production, and for recombinant products. In brewing, yeast from a previous fermentation is often used to inoculate fresh wort. The harvested yeast is called the crop, and inoculation is called pitching. This practice is common in the UK due to excise laws. The problems with this method are contamination and strain degeneration, often seen as changes in flocculation. In open fermenters, the top layer of yeast is discarded and the middle skimmings are harvested for pitching, as they have the desired flocculation and are protected from contamination. The pitching yeast can be treated with acid or antibiotics to reduce bacteria. In modern cylindroconical fermenters, yeast collects in the cone, suffering from stress, which reduces its viability as an inoculum. Storing the yeast cold and without oxygen further complicates matters. A key physiological feature is the sterol level in the cells, which is only synthesized in the presence of oxygen, yet ethanol production requires anaerobicity. This is resolved by aerating the wort before inoculation. Due to these issues, yeast is rarely used for more than 5 to 10 consecutive fermentations. Pure inocula are prepared using a propagation scheme with a 10% inoculum volume at each stage and continuous aeration, producing sterol-rich yeast. Propagators can be single-stage or two-stage semi-continuous systems, though there is a risk of strain degeneration. For baker's yeast production, an inoculum is developed through many aerobic stages. The first 3 stages are aseptic to minimize contamination. The yeast is pumped or centrifuged between stages. The first 5 stages have lower yields as they are not fed batch, while the last 3 are fed batch systems for higher productivity. Thank you for joining this comprehensive overview of the upstream process in fermentation. We covered the critical steps from screening and isolating the right microbes, to preserving them, improving their capabilities, designing optimal media, and developing robust inoculums. Mastering these upstream stages is fundamental to successful and efficient industrial fermentation. We hope this session was informative.